Hello there, and welcome to Preprints in Motion, the podcast taking a deep dive in the fast-paced world of preprints.
Join us as we sit down with early career researchers, discuss their latest preprint, and find out about their journey through the muddy marshes of academia.
Hit that subscribe button, leave a rating, and find us on Twitter @MotionPod. Support us by heading over to buymeacoffee.com/preprints. But for now, let's get into the show.
And this week it's cephalopods and RNA editing, both of which are incredibly cool. We also had an amazing chat about our visions for academia, although I may have stopped recording the best bits. Sorry. There's two kinds of episodes we seem to do. One is, I know the topic quite well, so it's a really cool, I like to get into the discussion. And the other one is, I know absolutely nothing about what we're talking about, and I just learn a lot. And this is one of those ones, because I'm vaguely aware of some of the stuff we're going to talk about, but very superficially. So I'm in for a really good lesson today. So I'm quite happy. They're my favourite ones.
I'm excited about this. It'll be fun.
So no pressure, but I want to learn.
Okay.
So we start off in the same place with everyone, which is just an introduction as to who you are, and a bit of sort of background as to where you are, how you've gotten to where you currently are.
My name is Kavita Rangan. I'm a postdoc at UC San Diego, and right now I'm in the lab of Sam Rick-Peterson. And so the lab basically is focused on sort of like understanding mechanisms of intracellular transport and microtubule motor-driven transport, I guess. And so I actually joined Sam's lab specifically to do this project because I basically was working at the MBL in Josh Rosenthal's lab, and that lab studies RNA editing in cephalopods. And so after leaving his lab, I was still super interested in basically exploring kind of like the functions of RNA editing in these animals. And I thought that motor proteins basically would be just a really good model to explore the functions of recoding. And so so that's sort of like the premise, I guess, under which I joined Sam's lab.
I mean, I definitely understand why, because it is a really cool project. So if we start basic, for me, mainly me, could you give us an overview of what RNA editing and recoding actually is?
Yeah. So the type of RNA editing that I'm studying, I guess, is like adenosine to inosine RNA editing. So basically this is a process mediated by enzymes called ADARs, and they basically, through like hydrolytic deamination, convert adenosines in double-stranded RNAs into inosine. And this can basically have just like a ton of different functions. But one of the things is that when this happens in mRNAs, it can happen in either like non-coding regions or coding regions. And when it happens in coding regions, basically the inosine is read by the translational machinery as a guanosine. So it's kind of like an A to G change. And so that can actually lead to sort of non-synonymous codon changes. And that's sort of what's called recoding. And I guess this happens in, I think, like all animals that have been checked, I guess. But in humans and in mice, most RNA editing doesn't lead to recoding. There are some like very famous sort of examples of recoding events that are essential, actually. But yeah, the vast majority of editing is not used for recoding, it appears. But basically work from the Rosenthal lab and the Eisenberg lab at Tel Aviv University showed that recoding in soft-bodied cephalopods, so that's like squid, cuttlefish, and octopus, it just happens to like an extraordinary extent. So 60% of basically their transcriptomes are recoded, which is crazy. And most of the sort of like recoded transcripts harbor many recoding sites. And so basically this is like has the potential to just sort of really diversify the proteome. And obviously like one question is sort of like, what are these animals using this for? And is it actually important for sort of like generating phenotypic plasticity in different tissues or environments? And so that's sort of like one of the questions.
It might be one of my questions. I mean, 60% sounds like a huge amount. Are we missing a lot, basically? Because it's an extra layer of regulation, right? So we're pretty accustomed to the proteome modifications. We're accustomed to genomic modifications, but I think that RNA bit is a bit, because I only became aware of it like in the past 3 years. So it's probably really old. It might not be. But, how much are we missing if there's that much of a genome being edited?
RNA editing is usually found through RNA-seq. So basically you have these like short reads and you sequence, and then you basically look for kind of like effectively mismatches between like a DNA-seq and the genome sequencing and then the RNA sequencing. And so that means that for a given edit site, there's sort of like an edit percentage, like a percent editing at every given site. So some sites are like, ubiquitously edited, but others are, maybe less edited. And then some of them are very low levels of editing. And so there's kind of this question of like, when RNA-seq is done in sort of these like complex tissue samples, for example, like, there's kind of this averaging problem. And there is kind of been this like interesting idea that maybe on like a single cell level, like editing might even be binary or something like this. So in terms of like, what is the true sort of extent, at least in sort of the work that I've been doing with squid kinesin, you can see that like edit sites that have very low percentage at one temperature, for example, have very high percentages at another temperature. So it seems like something that we shouldn't necessarily like say it's not important because it's a low level edit site, for example.
So what led you to be interested in RNA editing? Because I very nearly a few years ago wrote a grant about RNA editing and rapidly gave up because it was too complicated.
Yeah. Honestly, it's just the exact thing that I said. I just think it's like extremely fascinating that it's introducing so much heterogeneity. And I guess at just some very basic level, it's like, what is it doing? And I think one of the kind of ideas that I thought was really cool was the idea that like, okay, if you take the hypothesis and say it's true that, really these are functional variants like that are used by these animals, then it's kind of powerful because now you're saying like, if you look at a really highly conserved protein, you're saying like in humans, we're trying to understand how this protein or how this machine works. But now we have this organism where, here are all these functional variants that it uses to sort of modulate the function. So it's almost like showing how like nature or evolution, maybe it like is tuning the function of a given protein. And then you can kind of use that information to learn more about sort of like conserved aspects of that protein function. And so that was sort of like an idea that I thought was kind of cool and it's like, people, they might look at like disease associated mutations with like sort of the same lens, or they might use, like just sort of homology and just be like, oh, these areas are conserved across phyla. And like, so that might be interesting, or this is divergent. So that might be interesting. But this is just like a different way of that you can sort of say, wow, this, clade of organisms has all these functional variants and maybe we should just inspect what they're doing.
I mean, in terms of tuning protein function, are we talking making a protein more or less active, or is it a different protein?
Yeah, I mean, I guess honestly, it could be like anything, right? I mean, in the case of these motor proteins, I mean, I ended up looking at motility just because that was like a super quantitative thing that I could look at. But, other aspects of like, for example, protein stability at different temperatures or something like this is also probably really important, or how it interactions with other parts of the complex, for example. And so, yeah, this is sort of, I'm realizing I'm being meandering, but one thing that I think is cool and that I'm sort of starting to do is like, we have so many labs basically, or solving these like really high resolution structures of, for example, like the dynein motor protein complex or like kinesin protein complexes or whatever. And they're super like dynamic complexes. And we have all this like really high resolution information. And so basically I'm trying to build like a map of all the recoding sites across all the components of the dynein transport complex to maybe see just like, if you look at that, do regions pop out basically like that highlight sort of like protein interfaces or domain interfaces or whatever that might be interesting that we haven't really looked at from just using sort of like traditional structural approaches before.
It sounds like so much bioinformatics. I think that's why I stopped writing the grant.
I'm kind of just doing it like by hand, I guess, a little bit.
So could you give us an overview of what you did in this work and sort of your main findings, or what you think your main findings are?
Yeah, so like I was saying, I kind of came at it from like wanting to touch on or like explore, I guess, like two questions. One of which was like, can we use these recoding sites as like a map, sort of like I was saying, to sort of guide like the functional characterization of like highly conserved proteins? And then the other thing is sort of like, what are these animals doing with this crazy thing? And like, is it responsive to the environment? And like, how might that be used to change protein function? And so, yeah, so I ended up looking, I guess I didn't really go over this, but like, if you just look at if you do like a GO term analysis of edited proteins from squid, proteins involved in like transport intracellular transport and localization basically are enriched. Which probably just like reflects that this is like a highly regulated process, and like editing might be one thing that regulates it. But that sort of led me to sort of just look more specifically at editing of dynein and kinesin, which are really good models for like looking at dynamics of protein function. And they're really heavily recoded. So like dynein, I looked across. So this is data from basically, So another cool thing is that I basically just used data from another paper, which was from like the Eisenberg and Rosenthal lab. So like there's like a Cell paper from 2017 where they sort of characterized the sort of like editomes of 4 different species of cephalopods. So they looked at Octopus vulgaris, Octopus bimaculoides, Sepia officinalis, which is like cuttlefish, and then Doryteuthis palei, which is a market squid on the Atlantic side. And so they sort of published and made like really accessibly, I think, which was really nice, like this sort of editomes from these animals. And so just sort of like mining that data that already exists, you can see that like, dynein and kinesin, for example, are just really heavily edited. There'll be like hundreds of edit sites across these proteins. And they're dynein, for example, is a massive protein. It's like 4,600 amino acids, but still. So that's kind of just like amazing. And I wanted to basically first start off by saying like, okay, if we were to use this as a map, like, we kind of want to look at the intersection of residues that are highly conserved and residues that are recoded in these animals. And then we want to ask, if we make those recoding site substitutions, does it even do anything? Are these even functional? Or is this just kind of like you would see no effect? So the way that I was assessing function is just by doing like single molecule motility assays. So for both dynein and kinesin, I sort of used kind of like these truncated model versions. So for dynein, yeast dynein, you can have this sort of like minimal truncated motor domain that alone is precessive. And then for kinesin, I used human kinesin, which again, you can sort of use like a truncated construct that dimerizes still because it has some of the stock and then use that in motility assays. And so I sort of just made these sort of like cephalopod guided sort of substitutions in yeast dynein and human kinesin, and then sort of looked at motility and single molecule motility assays. And then the cool thing was that like, these substitutions that from what we know about the mechanisms of motility, which we know a lot, there would be no specific prediction for a lot of these substitutions of how they might affect motility, but yet, we saw significant changes in motility. And so that was really cool. And it kind of like, I think speaks to just the fact, I think there's like a lot more mechanistic work to be done on that end, but I think it sort of speaks to just the basic premise that like, wow, these sites are pointing to things that we probably wouldn't have ever tested had we not looked at how these animals do things. And then the other aspect of the project was sort of like, what do these animals use recoding for? And here I kind of ended up focusing on temperature. So like I did work with squid kinesin, and the squid species that I used is like this, it's called Doryteuthis opalescens. It's sort of like the market squid on the Pacific coast. And these animals, they have really like broad migratory, so they basically migrate from like Baja California all the way to like Alaska, like British Columbia. And so they are, there's a lot known actually about where they can be found because obviously like the fisheries are interested in them. So it's kind of cool. You can look through like, there's a lot known about, where they are at what time of year and things like that. So these animals, they do these just sort of like depth migrations. So they'll come up to the surface at night and then they'll dive deep. They can apparently, like, there's this really cool data from like Monterey Bay where since like 86 or something like that, they have these like remote operated submersibles that have been like tracking squid sightings, I guess, at depth, and then also measuring the water temperature. And so that's like, the crazy thing is that these animals are found at like even 1,000 meters deep. And so, they're basically experiencing really broad temperature changes. So, they can be found in really cold waters, like down to, I think, almost 5C. And then, at the surface, like, when it's warm seasonally, 15 or 20C. And so, so like the idea is like, temperature, I guess, is just like this pretty relevant factor for these animals. And in the case of kinesin, I think this is really interesting because human kinesin, it slows down markedly as temperature goes down. And it also runs like shorter. So basically the run lengths are shorter. And so like just a really naive question is just like, could this recoding actually sort of help compensate for what we know happens in human kinesin? And so basically I guess there was still sort of the outstanding question though, like, is recoding in these animals even responsive to temperature? And so UCSD works closely with Scripps Institute of Oceanography. And so the head of collections at Scripps, at SIO, Phil Zorowski, he basically dove down off of Scripps Pier and collected some Dorytoothless opalescens egg cases, and then brought them back to the tank room. And I was able to do some experiments with the hatchlings, which was really cool. And yeah, so like, I don't know, have you ever seen like sort of squid egg cases or anything?
No.
Cool. They're like, so they kind of look like these like long jelly fingers basically. And inside each of these like long jelly looking things, like there's many, many, many eggs, like hundreds, I guess. And so then you can see the developing like embryos, and you can see like as they develop sort of their eye spots and ink spot and stuff. And then they hatch out. And the animals that when they hatch out, they're like these, they're called paralarvae. They're really tiny. They're probably like a little over a millimeter long. And they have like, you know, chromatophores, and they kind of jet around. And yeah. And so as soon as they would hatch out, I would transfer them to tanks of like defined temperatures. And then after 24 hours, I basically just collected the animals and like flash froze them and then processed them for like RNA. I killed them. Anyway.
Yeah, it started off so nice. Such a beautiful picture for everyone. And then we just freeze them, kill them.
But yeah, so basically when I did that, I looked specifically to see, you know, what does editing of kinesin look like when you expose animals for just, you know, 1 day to these different temperatures. And then you can see, which is kind of cool, that editing increases as temperature decreases. There's many, many sites across the whole kinesin motor domain, for example. And so one question is like, what sort of variants are generated? Because, you know, it's not just one site. I wanted to make like sort of a real variant, for example, that would be found in the cold versus not found in the warm waters. Yeah, it's kind of interesting, but actually this is like not trivial. So to do like, basically the way that I did it was really old school. I just extracted RNA, made cDNA, and then like cloned it out effectively to basically get like single transcripts, and then used like, you know, whatever regular Sanger based sequencing, you know, to isolate each sort of transcript, in this case clone. And so with that sort of method, I was able to sort of see the distribution of edited variants, I guess, at 6 degrees versus 20 degrees. And then I made sort of these variants. I made like sort of like recombinant squid kinesin bearing these like different variants, and then tested its motility in single molecule motility assays at different temperatures. And so the cool thing there is just that the variants that were preferred in the cold basically sort of had increased landing rates and increased run distances at cold temperatures in single molecule assays. So this is like all sort of in vitro, but that was sort of what we were able to do. Can't do an experiment in the animals, but
I was kind of hoping, or in my mind, the picture of doing in vivo squid work was having these giant squid in these massive tanks, like something you'd see in the beginning of like a horror movie.
Yeah.
That's what I was kind of picturing.
Yeah, you know, the most editing happens in the Kraken. So like we had to only do it there. No, like, yeah, I mean, actually, you know, it turns out that the adults it's not trivial to rear them or even keep them alive for very long in a tank. So like the MBL has kind of like sort of famous for, you know, having regular access to squid, like the Atlantic Dorytoothless palei. But these animals, you know, once you bring them into the tank, even, you know, because they pretty much just die. You know, you can feed them and they'll live for a little bit longer, but they're not happy. So yeah, doing sort of like long-term experiments with the adult animals in lab is tricky.
Especially if they're big, they just eat you.
Yeah.
Maybe it might be helpful to refresh at least my cell biology. I see that as a cell biologist, it's embarrassing. Could you give a little bit of an overview as to what dynein and kinesin actually are? Just for anyone who's not aware.
Yeah. So dynein and kinesin are both motor proteins. They're microtubule motor proteins. And so basically, you know, one type of intracellular transport is like microtubule based transport. And so you have these microtubules that are sort of like polarized, and, you know, you have dynein, which walks towards the sort of minus end, which is oriented towards the cell nucleus typically. And then you'll have like kinesins, which most of the time walk towards the plus end, which would be like the periphery. And I guess one kind of interesting thing is that, like, for example, in humans, there are, I think, 45 kinesin family proteins, but only one cytoplasmic dynein. And so these proteins, you know, dynein sort of assembles as this like big complex, which involves like dynein, which is it's a multi-subunit complex, plus dynactin, which is another multi-subunit complex, and then an activating adapter. And then that sort of links it to diverse cargos and helps with trafficking. And kinesins also, they also have sort of there's a kinesin heavy chain, and a kinesin light chain, and then also cargo adapters. And so most cargos can be linked to both dynein and kinesin. And so you get bidirectional transport. And so it's a really complicated sort of process, I guess. And I guess pathologies and sort of both dynein and kinesin are linked to a variety of diseases and neurological disorders and things like this.
See, bringing cell biology, RNA editing, and squids. We're doing good so far. So you've mapped out all these recording sites. Is it possible to directly target a specific site of recording? Basically what I'm asking is, could you edit a certain site yourself, or do we not quite have those tools yet? Almost CRISPR for RNA editing.
You mean in squid?
Just generally.
Oh, yeah. So the things that you can do are that so a specific edit site, first of all, you could just change the codon to make it, quote unquote, the unedited or the edited version. But you can also sort of we know a bit about at least how, the sort of specificity, the substrate specificity of these ADAR enzymes. And so you could, I guess, in theory, sort of exploit that by changing nearest neighbor preferences, for example, or the overall structure to change, I suppose, an edit site, make it less editable.
Is this a case where it's specific targeted site editing that matters, or is it you have lots of editing across a range of sites and that's what results in change of function?
Yeah, actually, that's a really interesting question. So
we get those rarely, but we get them.
No, because so for example, with this combinatorial editing of the squid kinesin motor vein, not specific at all, there's 14 edit sites in this in the motor vein. And when I looked at sort of the combinations, it's not one of the cool things that I did actually with the help of my brother, because he's a mathematician at Current, although I don't know if he needed that much math skills to help me. Basically, he did this sort of pairwise correlation analysis to sort of ask the question of we see all these combinations, do any of does site 1 ever go, preferentially with site N, for example, and just to see do you actually see combinations that are preferred? And the cool thing is that you do, and you see it in in a tissue specific and sort of condition specific manner. So the pairs that go together in, for example, the stellate ganglion versus the optic lobe, those are 2 different tissues, I guess, cell populations. And then the combinations that go together at 6 degrees C versus 20 degrees C, those are different. I mean, there's a lot of overlap, but that you can see differences in the pairwise correlations. And so, but then, it's sort of in my mind, I guess, it almost seems too much of a grand design to say that any specific combination, that this is that the combinations are regulated in some way, especially when you sort of realize that for a site to be edited, there needs to be specific secondary, it needs to be double stranded. And there's so many other constraints, I guess is what I'm saying. That so in some ways it might just be a thing where, you get enough, this edit site and this edit site sort of have overlapping activity, and you kind of try to edit as much as you can. And that's sort of actually what I kind of saw, which is that when I sort of made the cold, so if you just look without looking at the combinations and you just look at these 5 sites are preferred in the cold, and then if you make those single substitutions in human kinesin, they kind of phenocopy each other. So any given one of them would increase landing rate or increase run distance. So it seems that was a trait or whatever that kept sort of happening, I guess, I don't know, in the cold. Yeah, or it was preferred in the cold.
Are the editing sites, are the sites close together, sort of physically, or can they be quite far apart?
Yeah, they can be really far apart. Yeah. So that's I guess one question is sort of especially for this combination stuff, is how is that sort of achieved? And I think that's a pretty open question.
So with the temperature response, because that was really, really interesting kind of response to get. Did you, or has anyone looked at other stress type responses? Because temperature in a squid is probably normal, but if you take cells out and put them to those extremes, that's a heat shock basically. So what about things like a nutrient deprivation type response?
So in cephalopods, no, or not that I know of, I guess I should say. Yeah.
Someone's done it, it showed nothing, we'll never know.
Yeah, I mean, actually, that in terms of the temperature in Drosophila, and also in like the species of like hibernating squirrels, people have seen that RNA editing on like a global scale, like sort of just changes with temperature, and that it also increases with decreasing temperature, and that's like one sort of very simplistic explanation for that is that you get more stabilization of double-stranded RNA at colder temperatures, I guess. But it's not that simple because, you do see things that are, warm preferred, for example. So there is some layer of regulation beyond just, an RNA in isolation. But yeah, in terms of like other kinds of things, I don't really know. I don't know.
Best answer. And how does the RNA editing machinery interact with other sort of regulatory components? So does it have to happen in bits of DNA that are accessible, so you don't sort of chromatin interaction, or can it happen?
Oh, in RNA you mean?
Yeah.
You can basically do sort of like editing reactions in vitro with like an in vitro transcribed transcript and purified ADAR. But the editing that you see in those like in vitro reactions doesn't fully recapitulate editing that you see in vivo. And so I guess there are other things that are sort of like either, stabilizing certain folds or maybe directly interacting that affect activity.
Do we know where the editing occurs? Is this occurring whilst RNA is in the nucleus, or is it when it's being processed in a ribosome?
Yeah, so actually this is kind of an interesting difference between, for example, squid and like human or mammals. So there are basically three ADARs, I guess. There's like ADAR 1, 2, and 3. And like ADAR 1 and 2 are both sort of like catalytically active, I guess. And ADAR 2 is basically or the idea for like human ADAR 2 is that it's like basically exclusively in the nucleus. And that's where like editing happens. And you can see that because it also happens like in, for example, pre-splicing. But the interesting thing is that squid ADAR 2 is cytoplasmically localized as well. Yeah, and so basically that kind of opens up a whole nother realm of how ADAR could be, like where it's editing and when it's editing and all of that. And so that's kind of like a pretty unexplored aspect, I guess.
Could you edit twice? So edit in the before splicing and then edit after as well?
Yeah. So for like squid ADAR 2, it's found in the nucleus and in the cytoplasm. Yeah. So and then also ADAR 1, I should say, is it shuttles between the cytoplasm and the nucleus, and editing probably happens in both compartments as well in humans. And the interesting thing is that for certain sites where it's been looked at, a single site can be edited by either ADAR, so ADAR 1 or 2. And so, yeah, there's just like so many layers of like potential regulation for this.
I have more questions about that, but I think we should move on in the interest of time.
Where do I find out about the different bioarchive licenses? This CC, BY, CDXY nonsense is driving me nuts.
ASAP Bio have a resource for that.
Ugh, that's your answer to everything.
That's because they have everything you need to know about preprints.
Sure, they probably have the basics, like info on the preprint servers, but what else is there?
There's so much more. Looking to post a preprint but not sure what different journal policies are? They have a collection to help you out with that. There are meetings around preprints and associated services. If you want to know how preprint adoption has changed over time, there's even a page on that.
And COVID?
They have a big section on preprints and the pandemic, plus some really cool infographics for communicating preprints.
And university policies? Surely they don't have that.
They collect uni policies where possible.
Okay, they do sound pretty impressive, but is it not a bit of an echo chamber?
It can be, but ASAP Bio also engage with people who don't love preprints and have concerns. So we had an excellent discussion on this very topic a couple of months ago.
Oh, is there anything ASAP Bio don't do?
Honestly, no, they're so nice over there. They were so quick to jump in and support this show. It's your one-stop shop for info on preprints and open science initiatives, so head over to asapbio.org to learn more and subscribe to their newsletter for the latest in preprint news. If you want a deeper dive into the world of preprints, then look out for the next recruitment of ASAP Bio Fellows.
So one of the things we ask everyone is, why did you preprint the work, and whose choice was it to preprint the work?
Oh, well, it was sort of like both me and Sam's. We sort of like both wanted to. I preprinted the work because I think it's just, extremely great and important for there to be preprints. I think, it's interesting because I don't know if this is true, but in my head, I heard this at some point, which was that like, if you look at, preprints versus like that same paper post peer review, they basically like don't change that much most of the time.
That would be my work.
Oh yeah, okay.
I contribute that. Yeah, so that is basically true. We did that with COVID preprint, and non-COVID preprint paper pairs. But there's quite a lot of evidence now showing the same thing, basically, all doing through sort of different methods. So yeah, preprints don't change that much when published.
Yeah, but I think the more important thing is just that, like, just accessibility of, research, and just sort of like, I think it's a nice forum to like share your work.
It got a fair bit of Twitter attention, but have you had any sort of benefits from preprinting?
So it's funny. I don't have Twitter. Sam has Twitter, and so she would take like screenshots and send it to me, and be like, "Good job." And I was like, "Oh yeah, thank you." So but I guess the ways that it has benefited me is like you, for example, contacted me.
Nobody said that yet. All our previous guests should be ashamed.
Well, actually, I may say that might be the only thing. So for me, that's like tops, Oh, and I was able to share my, I don't know. No, I could have done that anyway. I was going to say like my mom read it, but my mom would have read it anyway.
That's good. I don't think anyone reads my work. To be honest, I don't think I've ever read my published work either. I'm so fed up with it by the time it gets out. I'm done. It's out. I don't need to read it. If there's mistakes, somebody else can point them out.
Yeah.
So you had a baby right after you graduated, and one of the aims when we started the podcast up, other than just shining the spotlight back on ETRs, was to try and bring a better discussion around all equality, diversity, inclusion issues. But one of the things we can tackle quite easily here is, how do you find balancing a brand new baby with being a postdoc, which is really hard?
Yeah, so I actually don't have a brand new baby anymore. I have a 7-year-old, which is crazy. Yeah. So, but yeah, I've actually had, so it's interesting. I think, being in academic research, I guess it's one of the few jobs that has like so much flexibility. And I've been like really fortunate with all of my like mentors that have just been like really supportive, I guess, of like that aspect of, me being like a mom and also like someone who works in the lab. But I've also actually taken a lot of time off or like outside of science. And so basically my daughter's name is Fable. So after having her, my husband was finishing his PhD at Rockefeller. And so then after that, we moved back to California and kind of just like camped around for a while. And so that was cool. But then we ran out of money. And then I started teaching like evenings at Santa Barbara City College. So I taught like a class in molecular biology. Yeah. And then we ended up taking postdocs at the MBL. And then after a year there, we moved back to California. And I had another year where we were doing other things, and then I started in Sam's lab. And so basically, like to me, it was really important. I really wanted to spend time with my daughter. And so I basically just kind of tried to find ways that I could do that. And that sort of, I guess, ended up in being like, effectively 2 years of being outside of science and coming back, going outside and coming back, things like that. And yeah, I mean, it is tricky because, but this is just, there's school ends at, like 2:30 or something like that, and so, finding after school care and like all that, is just, it can be a lot. And summer is, I'm always just like, well, why don't we all have summer vacation? Like, how does this even work? So yeah, so it is tricky. But like I said, I think compared to most jobs, being a grad student or a postdoc, you do get a lot of flexibility.
It's also a really good example of how you can step away and come back. Because a lot of people, there's certainly all that sort of conversations across Twitter. A lot of people are quite afraid to step away from science because they're terrified they can't come back. But it is, and it's getting a lot more fluid, which is a good thing.
Yeah, I honestly think it's almost like I don't mean this in like a bad way, but our skills are so specialized, let's put it that way, that honestly getting a different type of job would be more difficult, I think. So yeah, I think it is like you become very specialized for academic research,
I've been looking around because I'm trying to get out of academia, and it is you very much have a specific set of skills that are really not that useful outside of science. All your transferable skills you get really early on. Like my postdoc, I get surgical skills. I never need to do surgery on a mouse again when I leave, but I can.
What you really want to just say on your application or whatever is I can figure it out probably is what I want to write down as my skill.
I'll learn as I go and I'll pick up real quick, I promise. So you also came from a C. elegans background, so you've kind of jumped, I don't know if we can call squid a model organism, but you've jumped across species. So how do you find that?
So actually I didn't come from a C. elegans background. Well, I mean, I did in grad school I did end up working with C. elegans extensively, but so I joined a PhD lab that was at Howard Hang at Rockefeller, and now he's at Scripps actually, but so he's sort of like a chemical biologist, and so the whole lab was sort of focused around using sort of click chemistry reporters to study sort of protein lipidation, sort of making new reporters, and then also analyzing different types of fatty acylation. And so when I first joined, that was basically what I was doing. I was looking at bacterial lipoproteins using click chemistry reporters. But then I guess I just started, and especially at that time too, I mean, I was, there's all this stuff coming out about the microbiota and, intestinal bacterial interactions. And I was really interested by that, but, basically dissecting sort of the mechanisms of how, intestinal bacteria interact, or the mechanisms of action of specific beneficial bacteria. It's really difficult because, just that milieu is so complex and So I wanted to sort of think about what would be a good sort of model system to kind of try and dissect that, and basically started reading these kind of interesting sort of anecdotal things about Enterococcus faecium. And so, Enterococcus faecium, I think I read this somewhere, but it's kind of almost like this primordial member of the gut microbiome, because you can find it in the guts of lamprey and snails and humans, and and then there was this paper from the Oswald lab where they basically saw that C. elegans, Enterococcus faecium could basically, if you feed C. elegans Enterococcus faecium, it sort of can reside in the gut of the worm, but doesn't cause any harm as measured by sort of survival. Oh, and then the other component to this is that, there were all these reports that Enterococcus faecium sort of could inhibit salmonella pathogenesis, in different dogs and in chickens and various animals, but nobody really knew why. And so I was could you recapitulate basically that interaction in a C. elegans and sort of ask if you sort of precede, I guess, the guts of C. elegans with Enterococcus faecium, does that inhibit salmonella pathogenesis? And then the cool thing is that it does. And so then that was a really nice little system where I could figure out that, how maybe this works. And then we were able to sort of do experiments in mice as well to show that, the same sort of secreted protein, which was a peptidoglycan hydrolase, that could inhibit salmonella pathogenesis in mice as well. And so, yeah, so that was sort of just its own world. And then when I sort of took off some time and took up time, taught, then went to the MBL. And then at the MBL, that's sort of where I was even just introduced into this whole world of RNA editing and cephalopods. And then so it's totally changed gears there. But yeah, for me, it's I guess I can sort of get interested in most things like this, I guess. Yeah. And my instincts are just sort of be oh, what would be a cool way to what would be a weird model to test this, or well, how could you, I don't know, that's sort of I guess I'm more of a generalist.
So we ask everyone about what we think could be improved in academia, which is one of my favorite questions because it means we get to go on a rant, and I love doing that. Because academia is quite hierarchical and there is a huge pay disparity, especially if you look outside of academia. Do you have any proposed solutions for tackling those issues?
Yeah, I mean, it is really interesting because I almost feel it's a thing where things keep what is that phrase, passing the buck or something like that? It's kind of at the end of the day, if this whole, endeavor of basic research was better funded, then I as the PI could pay you more and something like that. But I think I don't know, the way that I think about it is there's this idea that everybody wants to be this sort of island, or this sort of independent lab, and it's okay, I want to be able to do have the freedom to explore the questions that I want to explore. And so you end up making these super like resource intensive sort of like islands, and every lab is different. And I don't know if that's like necessarily the best way, but I do see how it does afford freedom, like, in some ways. But I think that it also like sort of creates sort of like more waste, but it also sort of it can breed sort of more inequality, I guess. Yeah, it is a good question. I don't know what do you think?
I don't know either.
Okay, can I tell you a weird dream?
Go on.
Okay. So I don't know. I have this idea that if I ever were to get a lab, I want to just basically be like, look, the easiest way to make this better is like, why am I giving a talk on shit you did? Or like, why aren't you writing grants for what you want to do? Like, it would just be like where we all facilitate each other in the questions and research that we're interested in. And there wouldn't be like a hierarchy. I think you could run a lab that way. And I think it would be really cool and probably really fun. And you'd probably, I don't know. I think, I don't necessarily see why people don't do that.
I mean, you'd probably have postdocs who are really skilled sticking around a lot longer.
Yeah, and then there's no reason to. Like, then it would sort of also get rid of this, like, island idea, right? It's like, okay, now we're just, like, a group of peers who are friends and we work on different things together in the same space. You know what I mean? And that just seems a lot healthier for, research and everything. I don't know. So I don't know. That's sort of, like, what I would want to do. But I don't know.
I think that's a good solution. It's a good way of doing, and you could do that locally. You don't need the whole infrastructure support to do that.
Yeah, exactly. And like the whole point is that like, I think if you just sort of don't have a PI be a gatekeeper to sort of like grants or, I guess just like how even just like you were saying, like talks and all these things, that would in of itself open it all up. And people who don't want to do that don't have to do it, right? They could be like, oh, I actually just prefer being like, more of like a technician role or whatever. But the people who want to do that would be supported.
Yeah, it's just about giving people the support they actually need, which is sorely lacking in most places. So I've just written a bit about what it means to value postdocs, because my most recent getting into trouble at work was I made comments about postdocs not feeling valued. That was it. That was my comment in a Nature article.
Oh, wait, really? And then you got in trouble?
Oh, yeah, they proved my point. It was great.
They were like, be quiet, you're not allowed.
So then I decided to go out and write a whole article all about it and really expand on that one sentence. But I want to follow that up with a sort of solutions article. So it's quite interesting to see what other people think as to how we could fix things. It's not an easy thing to do at all.
What do you think are like some of the solutions?
A lot of it is, or at least my focus is more locally, because I think the system's never going to change. I'm not that optimistic anymore. I think there's too much. Science is too career driven, and like the UK political situation at the moment, if you have people who are in it for their careers, they're not going to change a system that works for them. And so science as a system won't change until those people are gone. And sadly, those are the people we keep putting into fellowships and lectureships. So they're not going to disappear. So I focus a lot more locally, where you can make a difference. And it's things like you said, where you can run a lab differently, or where, and we see it certainly in the UK, we see this a little bit, where you get pairs of people teaming up to create a lab together. So you've got a joint lab, and each person's bringing their expertise, and then you've got more money, you've got more resource, you've got a bigger team potentially, and you can just do more and you can share more. So when I did my PhD at Sheffield, they were all different labs, but we worked really closely together. Not quite closely enough to share reagents like that, but we'd go on lab retreats together, and it was just, you had your lab, but you were part of a thing, and it made a huge difference. Whereas everywhere I've been since, you have your lab, and that's it. I've been some places where people don't want to help you, that blows my mind.
I know. It's so bizarre, because it's like, it's unclear to me, like, why. Yeah, like the whole point of this is just
careerism.
Yeah, like the whole point is to just like noodle around and be like, what is around me? Like, let me like, think about it. And yeah, it's to me, it's like sort of similar to writing a poem or something. It's like the same, a lot more resource intensive, but like the same idea is like, it's just a one philosophical approach to like understanding the world around you. And it's, the pinnacle of just being like a human. So I don't get why people try to like, I don't know what else, they're in it for the cash, man.
Yeah, they're really stupid then, aren't they? They're in their own career.
Yeah.
All right, I think that's it. I'm out of questions.
Thank you so much.
And that's the show, folks. If you enjoyed listening, then hit that subscribe button to get the latest updates straight to wherever it is you're listening. Don't forget to rate us on Spotify or Apple and follow us on Twitter at MotionPod. You can find links to things we've just discussed on our website, preprintsandmotion.com. If you'd like to tell us what you think, then send an email, shout at us on Twitter, or shout at us if you see us walking down the street. This has been a JMJ production, generously supported by our friends at ASAP Bio. Until next time, have a good week.